So after getting familiar with the interleukins, let's talk about cell surface proteins, and those are very much important regarding immunostaining and in sorry, and in rheumatology, and in a lot of chronic diseases, chronic ulcerative colitis, and chronic leukemias. We need to find surface proteins in order to find the etiology of the disease. So let's talk about cell surface proteins. First of all, we have to describe T cell. So in a T cell, generally we have first one TCR, T cell receptor. T cell receptor, as we have no, as if you watched my previous videos, we have described in our MHC lecture that TCR binds with MHC, major histocompatibility complexes, in order to uh, get presentation of antigen to activate T cells. So TCR binds with MHC of the APCs. Then we have on the T cell generalized on regardless of their differentiation, T cells generally have TCR. Then they have CD3, which helps in TCR to signal transduce to express MHC. So signal transduction, CD3 is important in signal transduction. Then we have CD28, which binds to B7 of APCs. So these three are the main general which are found on regulatory T cell, helper T cell, cytotoxic T cell. Next, we describe T cell cytotoxic T cell, which has the CD8 receptor. Then we have the regulatory T cell. Regulatory T cell is the successor, or is the byproduct of T helper cell. So regulatory T cell has obviously T CD4 because it is derived from T helper cell, and then it has CD25. After which we have the helper T cell, and which is Th2. It has CD4, then it has CD40 ligand, which helps to bind with CD40 in order to co-stimulate B cell. Then we have CCR5 or CXCR4 for HIV receptors. HIV, that's why HIV particularly affect the CD4, because they have the receptor on these cells, and we have to monitor the helper T cell CD4 in order to obtain the prognosis of HIV. After T helper cell, we have a hematopoietic cell, hematopoietic stem cell, and they have CD34. Then we have natural killer cells, they have CD16 and CD56. 56 is very much specific marker for natural killer cell, and CD16 is used to bind the FC portion of, it's like phagocytosis, so it used to bind FC portion of the IgG for phagocytosis or complement fixation. Then we have macrophages and B cells. For the B cell, first of all, B cell have the B7, or we can also say that CD40 or CD80 or 86. The B cell B7 binds to CD28. Then we have B cell MHC2. MHC2 binds with the CD4 and CD40 and 40L. Then regarding CD40L, we have CD40 receptor. Then we have the B specific, that is B19, 20 and 21. Then we have IG for antigen binding receptor. So this is 1 2 3 4 5 6 7. I think I have discussed its all receptors, after which we have macrophages. Macrophages also have MHC2 receptor. Macrophages also have the B7, but in this case they have the CD80 and 86 receptor. BC for in order to bind macrophages and T cell interaction, we have B7 to CD28. Then we have on macrophages we have FC and the C3b receptor for phago receptor for phagocytosis. And after which we also have, like T helpers have, we also have CCR5 for HIV infection. So in macrophages, in our innate immunity, we have CD14 for LPS and LOS antigen binding, and this is also known as toll-like receptor, which activate different cytokines. And this thing we discussed it in the innate and adaptive immunity. So this is all about and the last one is CD40. So this is all about the receptor. Once you get memorized, try to make conceptual understanding of these receptor and the interleukins, because when you conceptualize all these receptor, what are their functioning, try to connect one thing like the receptors with their activation, what their function. Try to spend a few minutes on this topic, and you will memorize, you will understand all the receptors, all the surface markers, all the interleukins, all the cytokines very much easily, because these all make senses. Like in MHC lecture, we know that TCR in the CD3, in TCR we have a CD3 interaction. That's why we know that T cell, yes, T cell has CD3 and TCR. Without this, they cannot activate it, or they cannot function properly. Then in our lecture of T cell and B cell activation, there we have discussed the B7 and T22, CD28, cytotoxic and helper CD8 and CD4. We have discussed this in T cell differentiation and also regulatory ones. Then CD40 ligand and CD40, we have discussed this in our lecture of T cell and B cell activation. This is an additional information, but yet it's understandable. It's linked from HIV topic to this CD4 and their significance. This is a new thing which we have to memorize it by heart. Then we have natural killer cell and this receptor we have to memorize this. But whenever look on this FCIgG, then we understand that okay, natural killer cell complement fixes the complement in the innate immunity. So conceptually this is understandable that for complement fixation we have the FC portion of the immunoglobulin. So in order to fix that complement and macrophage. For macrophages we have this this CD14, C3b and FC portion. And after which we have the B cell, again the same story, but in this B cells are the APCs. That's why they have Ig antigen binding site. So without this site, the B cell can never be perform their function as a antigen presenting cell. Then these three receptors are the B cell specific receptors. Then CD40, it is from our previous lecture from T cell activation and B cell activation. Also the macrophages, we have discussed macrophages and their FC and C3 binding in the complement pathway as well as in the antibody structure, immunoglobulin structure. From there we have known that the macrophages bind to the FC portion of the immunoglobulin. And afterward, in our innate immunity lecture, we have known that the LPS and LOS bind to the TLR receptor 2 of the, which is also known as CD14, and CD40 and CD40 ligand, they are present in the interaction of T cell and B cell. So if you just these all are the high yield topic and high yield points on the USMLE, and whenever you make links from one topic to another, then things will be very much understandable, things will be very much easier for you to understand, and it remains in your mind post test period. Like in your exam you have the full concept of this thing, but after your exam you keep on reminding these things, because these all are interlinked. And in my previous lecture I have discussed that like an orthopedic surgeon, he knows everything about bones, and the understanding of the bones is a very hard work, and that's why he paid a lot of money. The hospital pays him a lot of money. But in the rheumatologist, he have to know a lot of interleukins and cytokines and these receptors, and on a whole immunology. So all the hard work which spent on these understanding of interleukin, how they function, how they interact, that makes him a lot much more expensive than an orthopedic surgeon. So that's why these things are very much have their importance in our future life. That these things decide whether you are going to be an orthopedic surgeon or you are going to be a rheumatologist, because the hard work on whatever you think, whatever thing you do some effort on it, and it will give you a lot of successes in your life. So that's a natural rule. So in next lecture we will discuss the immunity section.